for those of you in the audience, if you're joining us today, you're probably either already an NF Core or Nextflow user, or you've probably heard of them, have some interest in working with these tools, and you're curious to learn more about how to get started quickly and easily. So let's start with what exactly is NF Core. On the screen, this is the tagline that you'll see when you go to the NF Core website. It is a community effort to collect a curated set of analysis pipelines built using Nextflow. So of course that leads us to the question, what is Nextflow? Nextflow is a workflow manager that allows you to chain together different steps into a scalable and reproducible pipeline. Nextflow is very popular amongst the bioinformatics community because it is able to handle the complexity and parallelization required in many modern bioinformatics analyses. So you can see on the screen here some key features of Nextflow. I'm not going to cover any of this in depth, just going to mention that Nextflow is a very powerful tool for building and running pipelines. Now, back to NF Core. The NF Core community provides essentially a set of guidelines that are the best practices that the community adheres to. So these are going to be guidelines to benefit both facilities and downstream users of these pipelines. So things like ensuring that there is always proper documentation and ensuring that these are validated pipelines that you can use and trust and reproduce. These are also best practices for the developers of these pipelines. So companion tools, automated tests that every pipeline has to pass for validation and release. And because of all of this, we can say that these are indeed best practice pipelines for the community. For the pipelines themselves, the NF Core community strives to collect a curated, that means minimal, set of pipelines. So there is one per data type. For example, you will only find one RNA-seq analysis pipeline, but you can run it in a lot of different ways. All of these pipelines are configurable with many, many different parameters, and you'll also find multiple release versions, so they've all been updated along the way. One of the main benefits of these pipelines are that they are portable, they are reproducible, and they're open source. Now, to say that we're big fans of this here at CodeOcean is a bit of an understatement. All of these software and dependencies that go into these NF Core pipelines are packaged together so that every pipeline is guaranteed to run the same way every time, no matter where you are running this pipeline. Everything within NF Core is held under an open source MIT license, which strongly encourages reuse. You can take a pipeline, run it multiple ways. You can build upon it, make it your own. It's all highly encouraged and easy to do within Nextflow itself. And here on the screen, we have the actual number of NF Core pipelines. As of today, NF Core has 60 released pipelines and 34 pipelines under development. So it's clear that the NF Core community is growing. The number of pipeline contributors and pipeline users are growing, and here at CodeOcean, we're interested in making it easier than ever for our users to access and run these powerful pipelines. So now, before I move on to the next segment where we dive into how we make that happen, we're going to pause for a quick audience poll just to get a better understanding of the expertise and interest of our audience today. All right, and we'll go ahead and close the poll. Thank you everyone for participating. It looks like about a quarter of the folks in the audience are proficient in Nextflow and comfortable using NF Core pipelines. Another quarter say that they are somewhat familiar with NF Core and these pipelines. And about half are here to learn how to install and run NF Core pipelines. No one responded that they're using Cicera or Nextflow Tower. So thank you everyone for participating. These results are really interesting to see, and I hope those of you who are here to learn how to install and run these pipelines are going to find the session really helpful. So let's go ahead and move on. We've talked a lot about NF Core and how great their pipelines are. Before I actually show NF Core and CodeOcean, I'm gonna briefly discuss my own experience running these pipelines outside of our system. And to just give everyone some context to this, a little bit about my background. I got my PhD in molecular biology, While getting my doctorate, I did both the wet lab experiments, so I worked with several mouse lines. I did sample prep, library prep. I also did the dry lab sequencing analyses. I would definitely say that I'm more comfortable at the bench. Computational work is not really where I excel. And I think at the time, the struggle was really more due to not having the right tools or knowledge to really go above and beyond what was absolutely required to analyze my data. So now, fast forward a few years, I had taken it upon myself the challenge to check out NF Core because I'd heard a lot about the pipelines, and of course, I started with their most popular, which is RNA-seq. Now, I've analyzed RNA-seq data before. I am familiar with the classic tools, FastQC, Trim Galore, CutAdapt, STAR, etc. I didn't think this would be easy by any means. It definitely brought back some grad school flashbacks for sure. I will say I have no Nextflow experience, by the way, but I know that NF Core provides extensive documentation, so I was ready to give it a shot. And this is what it kind of looked like for me. I was just doing this on my own personal laptop. We start with the software requirements. Of course, you will need to install Nextflow, and then you will need one of the following software packaging tools, either Docker, Singularity, or Conda. Here at CodeOcean, we like Docker, so that is what I used. And then you will need to fetch your pipeline. Now, Nextflow will do this for you. You can specify a version number of that pipeline for reproducibility, which we also like here at CodeOcean. Every pipeline comes with a small test data, so it's good to run that just to make sure everything is working properly. And lastly, you will want to run it with your real data, which often involves configuration files and parameters, such as what you're seeing on the screen here. So that's basically the long and short of what I did. So it sounds pretty simple, right? What exactly did I learn from my experience? So these are really just my own takeaways and recommendations. This is obviously going to vary person to person based on their own expertise and experience level. I would say some minimum requirements. Certainly what would be helpful to new users would be having some general scripting experience. You should also probably know your way around command line. That's where you'll be doing the installation. Having a foundation of Nextflow is incredibly helpful, and this is definitely something that I did not have. And actually, that's part of what made the whole process a little bit intimidating. There are so many Nextflow training tutorials that you can find them on their website, you can find them on YouTube. If you're interested in learning Nextflow, I think just diving into tutorials and documentation, that is a good way to go. Having knowledge of the NF Core template, which is basically the layout of each pipeline. So every NF Core pipeline must be built using a specific template. This is to standardize the pipeline code structure. Having a good understanding of the structure, the directories, the files, modules, assets, it can be really helpful when you're trying to run it for the very first time. And then knowing about the NF Core tools. So these are the companion tools that are developed by the community. Here are some of them listed on the screen here. Knowing how to write a sample sheet. So for this, I basically just followed the pipeline documentation for this part, as the specifics of it really depends on the pipeline that you're working with. And lastly, there are the pipeline configuration files. So there is a large number of configurations. There's the default config files, which allow you to specify default resource allocation. There's configuration profiles, which you're seeing on the screen right now, for specific software packaging, for test profiles. There's also institutional profiles and local configs and many more that I'm not going to pretend that I'm familiar with. So some of this is going to depend on where you're running your pipeline. So whether it's on your local machine like I did, or on HPC, or on the cloud. So knowing which configs you'll need to use and set up and which you can just leave alone, that's definitely an important step in the process. You may even need some DevOps experience, or more likely the help of someone else to take care of the DevOps side for you. For example, to set up AWS batch configurations if you need to be running these pipelines on the cloud. So those are really just the basics. All in all, it took me about a week to figure out how to install and run this RNA-seq pipeline on my machine. Now, again, if you have a stronger bioinformatics background than me, or if you're already familiar with Nextflow, it may certainly go a lot faster. But basically, for me, it was a lot of poring over documentation, trying to figure out which configurations I need. And, if something went wrong, I would just ask someone with more Nextflow and NF Core experience than me, like Stephen on this call. So that worked out pretty well. And if all else fails, you can always head over to the NF Core community on Slack. You are sure to find your answers there. And now I am going to cover NF Core in CodeOcean. I'm just going to show the whole process in a demo on our system. So how to import your pipeline, how to run it, what a result looks like, and how you can integrate NF Core into your workflow. But before we do that, for anyone in the audience who isn't familiar with CodeOcean, I want to give a very brief introduction on who we are and what we do. We are a cloud-based platform that serves as a centralized workspace for all of your computational science, as well as a repository for your computational assets. We provide self-service automation that mitigates DevOps by making it easy to provision and scale cloud resources. An analysis built in CodeOcean is traceable and reproducible. That means it's going to run the exact same way 10 years from today, and you will always know how a result was generated and how to reproduce that result again. Collaboration in CodeOcean means sharing analyses and results with the click of a button. We support IP protection for proof of invention with automated lineage and provenance. And lastly, we are built around FAIR principles and designed to maximize the reuse of analyses and data. So you won't ever have to rebuild from scratch again. And with that, let's take a look at how we use NF Core pipelines in CodeOcean. Not that slide. Okay, let's hop over into the system. This is my CodeOcean pipelines dashboard. So these are all the pipelines currently available under my CodeOcean user account. By the way, this demo environment is currently running on AWS. Our system is always deployed into the customer's cloud environment, and our pipelines feature in particular runs Nextflow using AWS Batch, and we take care of the DevOps. So there's no need for users to figure out how to set up Nextflow and AWS Batch on their end. So let's go ahead and grab a new pipeline. Let me show you how fast and easy this is. Blink and you'll miss it, I promise. Let's go to new pipeline. We're going to click on import from NF Core. So this dropdown you see here shows every NF Core pipeline that is available at the moment. Let's go ahead and grab RNA-seq, the exact same RNA-seq pipeline I mentioned before. You can select the version number that you want. I'm just going to grab the latest and I'm going to click import. So what's happening right now on the back end, we are grabbing everything from the NF Core RNA-seq GitHub repository, and we are putting it into an organized CodeOcean pipeline user interface. This is going to take about, let's say, a few more seconds. And there you have it. So this is the NF Core RNA-seq pipeline. It is that easy. So on the middle of the screen here, we have the README file that comes with this pipeline. So it goes over everything from NF Core. So every step in the process, it includes information on how to set up your sample sheet, how to run the pipeline, and what your outputs should look like. So again, this comes directly from the NF Core community. On the left-hand side here, this panel, we have the app panel, which is a CodeOcean no-code user interface for running your pipelines and capsules. You can think of capsules as building blocks for our Nextflow-based pipelines. All of these parameters that you see here are very conveniently pulled from the NF Core pipeline. If this is your first look at an NF Core pipeline, yes, that's right. There really are that many parameters for you to customize your run. And our app panel now makes it easier than ever. So you can hover over each one of these parameters and see the information on that parameter as provided by the NF Core community. So that's the no-code UI for setting up your run, essentially. On the right-hand side of this capsule, this panel shows the timeline with the full history of the pipeline. It is also a UI for built-in version control. So let's say I'm gonna modify the title here. So just like that, the timeline is going to tell me that I have one uncommitted change here, and I have the option to commit it right from the UI. So let's go ahead, we can commit those changes. It tells me I've edited the metadata of this pipeline. So now let's go ahead and run this thing. So there are two things that we need to do before we can click the run button. We need the pipeline to be able to access our data, and then we need to tell the pipeline exactly what to run. So let's go into the Files tab here. We're going to go into Pipeline. Now, everything you see here is everything that was actually pulled from the NFCore GitHub repo. So let's go ahead and attach our data to our pipeline. So for this, I'm going to click Manage Data Assets. To run this RNA-seq pipeline, I'm going to need my reference sequence. I'm going to need an annotation file, and of course, I'm going to need my actual sequencing reads. So just like that, we can see my data is now sitting in the data folder. Now, my data currently lives in the cloud. I've set up these CodeOcean data assets so that my data can be easily attached to as many capsules and pipelines as I want, as easy as what I've just shown. Click Attach, and it shows up in the data folder. We can click Detach. Just like that, it's gone again. Let's get back our reference sequence. So another benefit of working with data assets is that you can also easily share data with other users, with even a whole team. There's of course built-in support for credentials, so everything is safe and secure, but then everyone can use the same assets instead of making their own. So next up, we need to tell the pipeline what to run. So we need to edit the sample sheet to change it from the test data that comes with the pipeline to our real data. So here's where knowing the layout of an NFCore pipeline is going to be helpful for you. I'm going to go into Assets, and this is the sample sheet that comes with the pipeline. I'm going to go ahead and upload my own. So we're going to click Done, and sample sheet demo. So this is now the sample sheet for the data that I have attached to my pipeline. All right, now we want to copy the paths to this file and to the reference and annotation file. So we're going to copy that into the app panel. So I'm going to click Copy Path to my sample sheet, hop over to the app panel. This is the input. Hop back to Files. We're going to grab the path to the reference, copy path, back to the app panel. That becomes the path to FASTA genome file. And again, if you need to know about what exactly these parameters are, that information is provided by NFCore. Lastly, we need the annotation file, copy path, paste it into GTF. And that's it. Those are the only changes I'm going to make. I'm going to run everything else with the default parameters. I'm going to go ahead and I'm going to click Run. All right, as we can see on the right-hand side in our timeline, we are already off and running. AWS Batch is going to schedule all of the processes for us. Now, this run is going to take about an hour, so we're not going to sit here and wait for it to finish. I have this exact same RNA-seq pipeline and the exact same run already completed in another tab here. So let's take a look at this pipeline. We can see at the bottom, this was first imported 23 days ago. This was imported by Steven. I can see that he made some changes to the sample sheet as well, and then the metadata. I can also see all of the runs and results. So this was the first run that he made, and I can see the parameters that he used to generate that exact result. So this timeline really comes in handy when you're collaborating with other users. And speaking of, I'm just going to quickly show how easy it is to share analyses in CodeOcean. So whether it's a capsule or a pipeline or a data asset, we can click Share and see all users or even groups who have access to this pipeline and the type of access that they have. So right now, I'm just an editor of this pipeline. I've got editor permissions, but not owner permissions. So we can go ahead and add another user. So let's add our colleague, Emily. and I can specify exactly what she can do with this pipeline, and I can give her editor permissions. And just like that, Emily is going to have access to this pipeline. So she can come here, she can click run, and it's going to run the exact same way for her as it does for me and as it does for Jake. No additional setup required on her end. If we wanted to remove access, it's just as easy. So I'm gonna quickly go over the output files of our run. So that's what we see in the timeline here. FastQC is just gonna give you a quality control report for each one of our samples. MultiQC is going to condense all of them into a single report. I'm gonna skip down to the star salmon folder. This is where we're gonna find the bulk of our analysis files. So bigwig, those are your normalized alignment files. You've got DEC2 for differential analysis, feature count matrices, etc. If you go back to the README file as provided by NFCore, they provide links to more detailed information about each one of these output files and reports. There is also a pipeline info folder. So we can click here and see these are all of the parameters that were used in this run. So this is the exact same list of parameters that is provided in the CodeOcean timeline. We will also see here, this gives you the software versions that are used for every tool and every package in this pipeline. And then lastly, in the Nextflow folder here, I'm just going to show the Nextflow report. So here we have basically a summary of the entire execution. We have the actual Nextflow command, which is what we see in this box here. There's also some general information about the run, such as CPU hours, directories. We also have some resource usage graphs, CPU, memory, job duration, etc. And then at the bottom, we have a tasks table. So we can also see here some of the tasks were actually cached. That means that we were able to use the cache from the previous run because there were no changes made. So this actually uses Nextflow's resume feature, so it's fully supported in CodeOcean. I'm going to quickly show the pipeline settings. So by default, we have it configured to use the cache from the previous run. So when I ran this, it was actually using the cache from the previous run that Stephen made. By the way, on this same settings, we can actually change the version of Nextflow. We can select a different IAM role, even a different error strategy. So it's very easy to customize your pipeline run. Going back to resume, let's say I wanted to resume from a particular run, we can also do that from the timeline as well. Just click the drop down and then resume run. So another thing that I did is created a result data asset from the star salmon forest. So that's what we're seeing at the top of the pipeline here. Now this does several things for us. I now have the folder, all of the files in it. This result now comes with full data provenance and lineage. So let's say that this result is significant enough to share. As a data asset, I can now share this with my colleagues, with other users. This data now has access permissions. So just like what I showed earlier with capsules and pipelines, I can share this with others. They can come here, browse the result files, and they can also see the provenance, exactly how this result was generated. So they can see the pipeline that I used, the 3 input data that I used, all of the parameters that I used for my run. And if they wanted to, they could reproduce all of this with the click of a button. Now, this data asset can also be attached to other capsules and pipelines for further downstream analysis. Let's take a quick look at the lineage graph that is automatically generated by CodeOcean. So we have here our NFCore pipeline. We also have the 3 input data that went into this pipeline. So this is a pretty simple graph. Let's take a look at another example. So this is a real result that involves, you can see here, a capsule whose output became the input for another pipeline, as well as there were 4 other data assets involved here. So you can see how very quickly it can become difficult to track where data came from and how results were generated. With our automated lineage and provenance, we do all of that work for you. This result is actually guaranteed reproducible. That's why it's colored green. And we can hover over any one of these nodes. These links are going to take us directly into that asset, just like that. And by the way, this is actually another RNA-seq pipeline that was made in CodeOcean. So this was not imported from NF Core, but it's super cool, so I have to talk about it. We can see here we call this a builder UI, we can see how capsules and data assets have been strung together to make this pipeline. We've got our app panel here that looks very similar to what I showed before in NF Core. So these are all of the parameters, and we can expand each one of them to see all of the parameters as specified by the creator. And what's awesome about this pipeline is we can go into files and I can show the main.nf script. This script was actually automatically generated for the user based on what they did on this UI here. So all of this Nextflow code that you see in this file, it was written for the user. So you can build your pipeline using this UI, and the Nextflow script is written for you. So now you don't need to know Nextflow to use Nextflow in CodeOcean. And with that, I'm going to end our demo there on a high note and hop back to the slides. We're going to start to wrap things up. So in conclusion, some benefits of using CodeOcean for pipelines. As we saw, you can import NF Core pipelines easily with zero software installation, no DevOps configuration, no reading documentation. Just select your pipeline from the dropdown in CodeOcean and click import. Your NF Core pipeline imports with a no-code UI to parameterize your run, which is certainly a lot nicer than digging through config files. You just attach your data and click run. AWS Batch is pre-configured and ready to go. No user level setup required. And if you're interested in building your own pipelines, we support a drag and drop UI that auto-generates Nextflow code. For all of the Nextflow experts out there, you can also just write pure Nextflow script in CodeOcean. We are, of course, more than just a pipelines tool. We are one platform for all stages of computational research, from your upstream data processing to pipelines to downstream computational analysis with built-in IDEs and apps running on the cloud. We automate environment setup with Docker built in, version control with Git, as well as cloud management. With our automated lineage and provenance, we provide a scientific system of record that captures the entire analysis and data lifecycle. And last but not least, we are built with fair science architecture, and there is zero platform lock-in.